Hi class, this is continuation of our week number 8 recitation, and this is on unit 18, the fungi. So we have a lot of slides to cover here because I want to give you as much information as I can in these PowerPoint slides instead of going back and forth between the PDF, and it keeps us focused along this way. All right. So hopefully we cover as much as we can without overdoing it. But I think, the more information you have, the better it will be for you. All right, so we're going to do our unit 18. So the fungi, what do they, how do they get their food? So they're heterotrophic, and some of them are saprobic or saprophytic, which feed off, dead material. And some are parasitic, which is not good. And some are symbiotic, which is very good. So here, how do they get their food? What they do is secrete digestive enzymes, and then they absorb the nutrients into the hyphae. So we'll talk about a hyphae later on, which is a thread-like structure, and they will form mycelium, which will bear into the fruit and body of the fungus. All right, so here the cell walls are made of chitin. So this is the structure of chitin here. And also we have to know that the cell walls of plants are made of cellulose, and also that we did the protisomycete, which is the water mold. Their cell walls are also made of cellulose. All right, so the same as the water mold. Okay, so now let us look at our phylogeny of the fungi. So coming down from the common ancestor that gave rise to the microsporidia, the chytrids, the zygospore fungi, which are the zygomycota, the bustular mycorrhizal fungi, which is the glomeromycota. And then we have the two other groups of fungi, which are called sac fungi and the ascomycota. And the other one is the club fungi, which is also known as the basidiomycota. These two groups of fungi, two lineages, are dikarya. All right, so that means they have two different types of nuclei. Okay, here I have put it out in regular format here. So this is same as we see in the former slide. Okay, so now let us look at the characteristics here on the fungi. All right, so this is very important that you do know the characteristics, so it helps you sort out the groups of fungi. Okay, so you have the different types of stuff you look out for, like the type of hyphae, the characteristics of the hyphae, what kind of reproduction they have, whether it's asexual and or sexual that we know of, and the representative species. So right here, we can see, for example, we will not talk about microsporidia as much. We won't even talk about it in this recitation. But however, we will talk of the others, which will be the chytrids, zygomycota, the glomeromycota, ascomycota, basidiomycota, and deuteromycota. Now, the type of hyphae, some of them could be aseptated, which means that they do not have any septum. So therefore, they are cenocytic. Cenocytic means that they do not have any cross walls, so they have multiple nuclei, more than one nuclei along the hypha or hyphae. Hyphae is the plural of hypha. And cenocytic, I say the same here. So they will not have any septation to separate the nuclei in the hyphae. However, in the ascomycota and the basidiomycota, they do have septation. So they are septated with different types of pore. So here in the ascomycota, we have a simple pore. And in the basidiomycota, we have a dolichopore. So it's a special pore in the basidiomycota. Some of these groups of fungi will probably just have asexual reproduction like Gloromycota, which is asexual, but it does not have any sexual spores. And also in the Deuteromycota, it shows that it has, so far we know of its asexual reproductive characteristics of having conidia, but no sexual spore has been observed for the Deuteromycota. Okay, and here we have our example as molds, and also we have penicillium. All right, and let us go on. So be familiarized with these here, because we'll talk about them as we go along, and I'll refer back to them. So here what we are talking about septation. In our hyphal arrangements, whether they have septation or not, we can see here. So this is aseptate, which means they have no septum to divide them. As you can see here, that they have septa here and septa here. So you can see that this is septated and this is septated. And here there's no septa or septum. So this is sinusitic. Figure 85A is sinusitic. B is septated. And if it has a simple pore, it could be Ascomycota. And here, this is a dikaryotic group of fungi. So it has two different types of nuclei. So you see a darker shade and a lighter shade. So this is dikarya. And that is C. So let us see what C says. So dikaryotic hypha with two distinct haploid nuclei. And here is septated. So the pore that's in our Ascomycota, when it's septated, it could be a simple pore or it could be a dolly pore. So figure 18-5D, you can see the septum here. And then now you can see the special dolly pore. So this dolly pore here is a Basidiomycota. Okay, so it's right, the Basidiomycete dolly pore. Okay, so what is the hypha? The hypha is one cell thick. It's more or less a tubular-like thread. It's haploid. And some of the hyphae, which is a plural of hypha, are sinusitic, which we talk about that they have no cross walls. So they're multinucleate. All right. And the nuclear envelope is maintained during mitosis. So the ones what we say which have septation, which are septate hyphae, they have pores in the cell walls. It could be either a simple pore, as in the Ascomycota. Let's go back here. We'll look at that slide again, which I'm pointing to here. Or it could be a dolly pore, as in the Basidiomycota. So here you can see Ascomycota. Does it say it here? Okay, simple pore in the hyphae, and here Basidiomycota is a dolly pore. All right, so this just describes it. So the main body of the fungi is called a mycelium. Mycelia is the plural, and this is a mass of a lot of hyphae. So one hypha will, and many more hyphae will make the mycelium, and many mycelium will make a mycelia. All right, so let us go on to the next slide. Here you can see that the hypha, the thread-like structure, and then they form this thick mat-like structure, which is called a mycelium. So here you could see how they are growing on a rotting log. And this year it's living off the dead matter of this decaying wood. All right, so let us look at the chytridiomycetes. Here we can see the life cycle of the chytridiomycetes. And let us look at the characteristic chytrids. They're aseptate, which means they have no septum. They have 750 to 760 species so far, and they're mostly aquatic. All right. And how do they reproduce asexually is that they have zoospores and they have alternation of generation. And the type of spores that say none, or they could have gametes or zoospores that have flagella in some of the chytrids. And our examples are allomycetes, and I'll show you some of the pictures that I have taken in the lab that we have prepared slides of allomycetes. So come on, let's go back to chytridomycetes. And this is our alternation of generation where it shows that let's start from the male mature sporophyte. So this is 2N. Asexually, they can reproduce the thin wall sporangium where they form the zoospores, which is 2N, because this is from the sporophyte. And then these zoospores will germinate to develop into young sporophyte. Okay, so then it goes here again in that cycle. However, they can also produce sexually. So here in our sporangia, which is 2N, they go through meiosis and they will make zoospores. All right, so the sporangia is resistant to, I guess, all kinds of conditions so that they can survive to make their zoospores. So once they go through meiosis, they will make these zoospores will germinate into gametophyte because the zoospores are N, it's haploid. All right, so it will develop into a gametophyte. So once it gets to an adult mature gametophyte, they can make, so you have female gametangium and you have male gametangium. So you see the female gametangium is on top and the one on the bottom close by, and they would make female gametes and male gametes, which will fuse syngamy. It will go through syngamy, plasmogamy and syngamy to form the zygote. And then once that zygote develops into a young sporophyte. So that is our life cycle of a chytrid. All right, so let us look at the other slide. This is one of the slides I have taken a picture of in class. So this is allomycetes, and it's so you could see the different stages of the allomycete. And here, since a lot of these chytrids are aquatic, with the global warming and the climate changing that's happening now, since we have more rains and all that stuff, in the tropics, they have a lot of frogs that die due to chytridal fungal infection. So this fungal growth will be on the skin of the frogs, and the frogs, they respire through the skin also, and they have lungs. So here they would get this fungal infection, and of course they get weaken and they die. So that causes a mass die-off of these amphibians, and it's not a good thing, especially now that we have to control, our greenhouse gases emission and all that stuff. So this is a bad indicator when we see a lot of die-offs of these amphibians. Okay, so let us go on to the next slide. So this is another simplified life cycle of chytrid, and it shows again how we're going to go. Let's start from the sporophyte generation here. So what happens is here in the myosporangium and the mitosporangium, that's what I call them. So this is in the sporophyte generation. They will go through meiosis to make myospores, and then once they germinate, they will develop into a gametophyte, and then they will make, when it gets to a mature gametophyte, they will make gametes. So gametophyte will make the female gametes and male gametes. And then once these gametes are released and they fertilize, so they go through plasmogamic procedure and syngamic procedure, where you will have a zygote, and then the zygote, once it's germinated, it will develop into a young sporophyte. So the whole cycle goes over again. So this is simplified also. So that was the chytrid. So let's go to the zygomycetes. The zygomycetes has a zygotic life cycle. Only diploid stage is zygote. And what happens here is only when two different strains come together can sexual reproduction occur. All right, so you can't have the two of the same strain. That happens. So here you see zygomycetes, sporangiophore, this whole structure that support the sporangium. And this is just other parts of our zygomycete fungal growth. So you have rhizoids, stolons, and all that stuff. So let us go on to the next, where we could see the life cycle of a typical zygomycete. So let us start where? Should we start here? Okay, so let's start in the mature gametophyte. So the hyphae are N. They go through plasmogamy or plasmogamy, depends on how you want to pronounce it. And then what it creates is this gametangia. And this, from the hypha, we have the gametangia, where it goes through plasmogamic procedure, and then it'll go through karyogamy, where it will form the zygote, because once the nuclei fuse, and this is where it is the diplontic stage here, where it is in the zygote alone. All right, as we were pointing before. And from here, it will develop a zygospore, and then from the zygospore, you'll have the sporangium that develop while it goes through meiosis. And then what happens is, once it ruptures, it will make the myospores. So once it goes through meiosis, from the 2N, it'll be myospores will be haplontic. And then once it germinates, it goes and develop into a gametophyte. It will germinate and it will make the hyphae, and then the whole cycle goes over again. So that is the life cycle of a typical zygomycete. Okay, so we did chytrid, zygomycete, and here. Now we're going to do ascomycetes, which are the sac fungi. So I got this picture here. And also, if you say sac fungi, it has a cup-like feature. So you see it's kind of cup-like. This is a nice blue fungi here that I put that picture. So it could be very attractive. And that they do have such a variety of colors instead of, your regular mushroom or other kind of fungi that you see. They're very colorful. And usually they said the colorful ones are poisonous. So, and some of them do glow in the dark. So what we have here is the other group of fungi, which is called ascomycetes. These are the sac fungi. Okay, so now they're called cup. Asco means cup also, C-U-P, like in a cup. So you can see these are cup-like. Now, this is the largest fungal group. And they do have sexual spores, which are born in these tiny sacs that's called asci or asci. And the singular is ascus. That's why we have the word asco. And that's why the tiny sacs, that's why they call it sac fungi. So where the spores are born are in these tiny sacs, that's called asci or ascus, as in singular. So that's where they're born. However, this forms a cup-like shape of the fruit and body. All right. So you have this interchangeable name of ascomycetes. And here the hyphae have septation with simple pores, and they do have asexual reproduction through conidia, and they're called conidiospores, and they're born on this stalk-like structure that's called conidia. Conidium is the singular of conidia. Okay, so here let us see what ascomycetes can do. It can it can cause infection in the tongue. It can cause, let's put this way, this person back then in the ergot, that when the wheat is contaminated with this fungal contamination of ascomycete, and it could cause hallucination and all kinds of stuff. So back then they used to have the witch hunt, and they would drown these people because they think that they are possessed. So in the ascomycota, we have morels and truffles, powdery mildew. So you get all these things. They are food spoilers. So if you leave food out in a while, they will get moldy and fungal growth and all that stuff. And it can cause really bad, I guess, can kill, and also can cause all kinds of other issues. Dutch elm disease, which means they're killing off all the elm trees, like in the States, and also they have chestnut blight. Yeast is also an ascomycota. All right, so let us look at the life cycle of ascomycete. This is taken from our PDF. So what we have here, let us start here. In this area where we have two different maiden types. We call them plus and minus. So in the hyphae, so the ascomycota, it is dikaryotic. So you have the plus type and the minus mating type. So the ascogonium and the antheridium of two different mating types. So once they get together, once they're ready to mate, they go through plasmogamic procedure here. So you have two different types. So let's call the nucleus here in the plus maiden type, the dark one, and the negative one as a lighter nuclei. So this, what would happen is when the ascogonium here, this is the view here. From here, you have these two hyphae that get together, that in plasmogamic procedure, you will have, in the process, you will have the two different strains. All right, so that's called karyogamic. Okay, so here you see it will give rise to this hyphae, which is dikaryotic, not karya, dikaryotic. So when you say dikaryotic, means two different types of nuclei, haploid nuclei. So we have two different strains that will develop into this ascus. So more than one is called asci or asci, and they'll form an ascocarp. So that's the cup-like feature here that you'll see, an ascocarp. So what it does is go through, after plasmogamic process, it will go through karyogamic process where the two nuclei fuse. And here as it fuses, you will see that you will have results through meiosis. And then, of course, this is showing like each ascus as they go through the different stages, but not all of them go through it at the same time. So here we see, as a result of mitosis and partitioning, so we'll start partitioning all, it will result in 8 ascospores. So the ascospores, once they are dispersed here, so coming down from that, after it develops, after these spores are mature, this ascus, the ascus will break open, each of the ascus will break open, but there are 8 ascospores that result from this meiosis. And then what happens is once they germinate, once they find a right condition for it to germinate, they will develop again into these hyphae. And again, the whole cycle begins. So that is a sexual reproductive cycle of the ascomycetes. However, they can reproduce by asexually. So asexually, they have this structure that's called conidiophore, and conidiophore has these like string of conidia on them. So conidium is one that has the spore, and this is the asexual reproduction. Okay, so hold on a second. Let me check to make sure that I did unmute. Yes, I did. Otherwise, it will be terrible if I was not recording properly. All right, so let's go back to the stage. Okay, what's going on here? Okay, so let's come back here. All right, so let us go on to the next slide. So here you can see, so now we have this ascomycota, which can capture, so this is like capturing a nematode, and it makes this loop around the nematode so that it strangles it, and then it exudes its enzymes to dissolve this nematode, and it absorbs the nutrients from the dissolved nematode. So here we see a nematode captured by this ascomycota, Arthrobotrys and Conia. All right, so this is our ascomycota. The next one we have here is the budding yeast, and this is Saccharomyces. And this is a picture of a slide that I had taken in class. So you see this is a yeast, which is, you could see the different stages of it reproducing asexually by budding. All right. So now let us move on to the next group of fungi. These are the club fungi. And it's called a club fungi because they have this club-like structure that's called basidium in its sexual reproductive cycle of its life. So this phylum here is a Basidiomycota, and it's called Basidio for the part due to its basidium structure. All right, so these are some of the examples we see here with stars and all that stuff. So now let us see some of the representatives of Basidiomycota. They could be mushrooms, puffballs, and they do have rusts that affect plants. Like here, you see they can be parasitic, and it will kill plants. So here what we have is Basidium is dikaryotic, the same thing as the Ascomycota, which means they have two compatible haploid nuclei in the hyphal cell. All right. So here, as we say, going back to the previous 3 slides, so you see how it's dikaryotic here, 2 different kinds of nuclei. And what else do I have to say for the dikaryotic hyphae? So here you see dikaryotic hyphae, and then it will go through its sexual reproduction. All right, so now, so this is the same for Basidiomycota. They have a dikaryotic hyphal cell. Okay, so here again, continuing with the Basidiomycetes and the club fungi. So I have to reiterate this, that it is dikaryotic. And here they have the special pore since they're septated. So you have septum dividing the hyphal wall. And they have a special pore that allows material to go between them. and communication. And that pore is called a dolly pore. All right, so this is a dolly pore here. So right here is the dolly pore in our Basidiomycota. Now, let us look at the reproductive cycle of Basidiomycota. So this here, which we will say the mushroom, the fruit and body, and which we call the mushroom. So you have the pileus, the cap, and you have the gills beneath the cap. So what we have is a simplified version that in the gills, these are the gills now that you see like this. They're all lined with basidia. So the basidia are these basidium here. So you see this club-like structure? Each of these here is a basidium. And collectively, plural is basidia, all right? So once they have gone through charigamy and all that stuff, they will make basidiospores. They will make 4 basidiospores, and when they're ready, they will be released. So this is a quick schematic of a Basidiomycota. So let us look at the other slide. Here in our book, figure 18-13, the Basidiomycete anatomy, and it shows the sexual reproductive cycle here. So here you see the hyphae here. So the dikaryotic, so they have 2 different types of nuclei. And then you have the stalk. So this is the fruit and body, the stalk annulus, and the cap or the pileus, and the gills underneath. So we take a little chunk of this, a cross section through the gills. And again, this is the basidia growing along the gills. Well, it's lined with basidia. And here is another piece of it here that's magnified here. So on the gills, so this is the gill surface. And then here you should see at the very end, basidium here. And then it releases basidiospores once it goes through meiosis and mitosis to form 4 basidiospores, and they would be released. And this is it's a bigger version of this part here. So here it would produce, what else? This is sexual reproduction. All right, so let us go on to the next slide, which give us description, more description of these. So as I would say, these club-like structures, each with 2 nuclei, that's why it's, and they are formed on the margin of each gill. So they go through nuclear fusion, which is charigamy, and then when they go through meiosis, and after they do the fusion, after they have nuclear fusion, which become 2N, they'll develop into this basidium, and then the spores will be born on its tip. And then it would show that these spores will be dispersed and then germinate. And then they'll start growing out into, well, it will be haploid, and then it'll germinate and have its, right here, germination. And then it will form into a mycelium from the hyphae. And again, the same cycle goes over again, where it shows that cytoplasmic fusion and all that stuff to give rise to this. Okay, so let us look at the next slide. And this, I have 2 red exclamation here, which means that you better know what this is. When you see this in the final practical, you know this is the gill. These are the gills of the fruit and body of a mushroom, of a Basidiomycota. And these things here, these structures here, these are basidia. And the basidia, which you could see also here, will make the basidiospores. So once you see this, you know it's in the phylum of Basidiomycota. And you know that it has a pore because it has septation. It's a dikaryotic group, which means it has 2 different haploid nuclei. And what else do we need to know? And it's called basidium because it's of the club shape structure that form from sexual reproduction. And also it reproduces asexually with conidia. So as say we have conidia, I have a picture somewhere. I'll have to go back onto the screen here. I'm forced to go back here to show you what conidia look like. If I could find it. Okay, I'm going through this. I thought I wasn't going to use this, but this is the only way I can show you guys. Oryzophus. I didn't show the other structure. That's Ascomycota. So please read your PDF. Go over it as many times as it takes for you to understand this. All right, so this here is asexual reproduction. This is for Ascomycota and also for Basidiomycota. But this one is showing this whole thing is for the Ascomycota. All right, so here, asexual reproduction, if you see this is a conidia. All right, Basidiomycota and Ascomycota have these reproductive structures. Let's see which other one has conidia here. Let's look. So conidia 2 also in Deuteromycota. All right, we have it Basidiomycota. We have it in Ascomycota. So these 3 have asexual reproduction of conidia. But also in the Ascomycota and Basidiomycota, they have also budding and fragmentation as forms of asexual reproduction. All right, so let us go back, continue with our slides. Which one do we have here? Sorry, we didn't need that. So we just finished talking about Basidiomycota. Now let's go on to the next group of fungus here. That's called Deuteromycota. This is the imperfect fungi. They have not discovered the sexual stage yet. So they haven't observed any sexual stage. However, what they have seen so far is the asexual reproduction by way of conidia. And this is what conidia look like also with a very good microscope. So this may be an SEM or higher magnification. And here they said, since they don't know what kind of sexual reproduction yet, but they say it's probably would be later classified as a Basidiomycota or a Asidiomycota. All right, so this is what we see like penicillin, and it's being cultured here in a petri dish of agar. And this is what, if you take a smear of this like in class, you'll see the hypha and the mycelium. So this matter is mycelium, and the darker areas, those are the asexual reproductive structures of conidia. All right, so here is a slide I did in class, penicillium, Deuteromycota. And here, if you make it bigger, you'll see the paintbrush like. But I didn't make it that big because it will take up the whole screen and you can't see the other structures of the mycelium of the Deuteromycota. All right. So now let us talk about the other group of fungus, Glomeromycota. So here we have, these are the mycorrhizals, that they have this symbiotic relationship between the roots of plants. And what it does is the mycelial and the hyphal connection here that this one is intracellular arbuscular mycorrhizal. So it is between the fungus and the root of plants. So this is one that goes intracellular. So it goes through the cells and they form this arbuscular kind of arbuscule or arbuscular structures. So these are the intracellular arbuscular mycorrhizal fungi because they are in the cell, in the cells of the root of the plant that they're associated with. And here you could see, like this is the root. Well, this here, all these here are the roots area. And the red that you see, the solid red, those are the hyphae of your glomeromycota fungi. All right, so here we have the arbuscule. And then what do they have here? In this here, they have a symbiotic relationship where the plant can get more minerals because it's limited in how far the roots grow. So what happens is for the hyphal growth and the mycelial growth, they have a wider area. So it extends its area in obtaining nutrients and phosphorus and all phosphates and all the other minerals that the plant will not get in its immediate area where as far as the roots and the root hair are extended. So it does expand its surface area and its reach of getting nutrients. And in return, the fungi would get the sugars, which is made, the carbon from the sugars that's made by the plant. So this is a symbiotic relationship. And this, the next kind of glomeromycota is the ectotrophic mycorrhizal fungi, which means it grows around on the outside, and then the fungal hyphal hyphae would go between the cells, so it can get its carbon and sugars. And this is called a Hartig net. So it causes a net like dispersal in between the cells of the roots of the plant. And what else do we have to say for this? So it's the same way, symbiotic relationship. So both can benefit of each other. And of course, there are other fungi which are parasitic, but not in this case with the glomeromycota. And here what we see is like the root of this sapling, how it has already established all this kind of mycorrhizal relationship. So all the other white fine hairs and this area, those are the fungal hyphal growth. And that goes to show how far the reach that wherever the hyphal growth are, they can get the nutrients, and that will be brought to the plant in exchange for the carbon from the sugars that's photosynthesized from this plant that it absorbed through the roots. So the root ball might be around here, this large, but the reach of this has extended much further over here. All right. So this is a symbiotic relationship. Okay, so let's go on to the next slide, which is lichens. So lichens, these are also symbiotic, where it's between a cyanobacteria or maybe a green alga and a fungus. So this here, now the fungal component of the lichen is the mycobiont, and it's mostly ascomycetes, and some could be basidiomycetes. So, but mostly in the ascomycete and And then you have cyanobacteria like nostoc. So this is a photobiontic component of the lichen. All right, so let us see the types of lichens we have. We have the foliose, which is leaf-like. This picture was taken from Queen's College on the last day of Queen's College before they had the lockdown. So I was on campus and I was trying to get pictures on campus of the different kinds of plants and birds and everything, the flora and fauna of Queen's College. And I happened to take this, and this is of a cherry tree near Powder Maker. Yeah, I could see Powder Maker in the back here. So this is a foliose, the one on the top, foliose, so leaf-like lichen. The next type of lichen is the fruticose. So you see these little red things like little fruits. So these are the fruticose lichens here. And then you have the crustose lichens, which are encrusted on the substrate, like on this rock. Let me bring my picture here bigger. Okay, so I have to make this a little so it could show more. All right, so this here is like encrusted on a rock. And the only way you can get this out is to scrape it out. So those are the three types of lichen forms. And now here we have a drawing of how a lichen could be, where the folious part, this is a folious lichen. So you could see the different layer, and you could see, you know, like how it is anchored to the substrate. And the green part is a photobiontic one, the algal layer. And then you'll have the other layer, which is the fungal layer. And here, figure 18-19, you see a cross-section of the lichen thallus. And then it shows the anatomy where the fungal hyphae forming the dense mycelium, and it attaches to the surface. And then these things, Soredia, once they pop out, these are asexual reproduction. So they will just, that's a reproductive unit. So here is a Soreidium is the singular of Soredia. So Soredia is many more than one Soreidium. And that is the asexual reproduction of this unit, fungal algal unit. And what else do I have to say for this? So it is also, and that will give rise to another lichen growing somewhere else once it's dispersed. While you read your PDF, you should see all this stuff here. So you got to read your PDF and go through this and come back again and look at the PowerPoints and hope it makes sense to you guys. All right. So here, this is one of the slides I've taken picture of. And this is the ascocarp, the lichen ascocarp. So this is symbiosis with ascomycete. Didn't say what kind of fungal group. Sorry, ascomycete. This is the fungal group. And then did not say what is a photobiont. So you see the ascocarps. This is like a cup-like structure. So that's why it's, you know, ascocarp. And then this part here, you'll see, if I had shown you in higher magnification, you'll see the other parts of it, like the photobiont part of it. But this is the best overall picture I can get, so it can show the ascocarp. All right, so now this is the end of our unit 18, and hopefully it's enough information for you guys to do well in your exam. And now what I'm going to finish off with is this video by a gentleman. He's not really a biologist, but he did this video, which is pretty neat. And his name is Stephen Axford. So he did this YouTube. Well, I saw it on YouTube. I don't know where he had posted it also. So this is really, really neat. He is really good at doing photography. So I'm going to end this recording by showing this video, and hopefully, well, if you don't want to see it at the end of this, it's fine. But this video is so good. You can look at it in your own time, but I will show it here, which is on YouTube that I have about to be set up here. All right, and it's half an hour long. Okay, so it's 32 minutes long. All right, so I'm gonna make it in widescreen, and I'll take off the headphone so that you can guys the video. Oh, voice. All right, so let's play it.
My name's Stephen Axford, and I take photographs of fungi. This stage I'm standing on is an endangered lowland subtropical rainforest near where I live on the north coast of New South Wales in Australia. Over the past 10 years, I've taken many photographs of mushrooms in this forest, and I've also taken photographs of mushrooms in some of the most remote forests on the planet. Photographing mushrooms has become a passion that has completely changed my view of the world. This is the first mushroom I ever photographed. Its scientific name is Cortinarius archeri. Why did I photograph it? Well, it was back in 2003. My wife had died 5 years earlier from breast cancer, and then I had a life-threatening illness. I'd survived both, but when you face death, it makes you rethink your life. And I was looking to reinvent myself. The wild places along the Australian coast and the ancient forests became a sanctuary for me. I was walking along a coastal track one day when I spotted this purple mushroom. I didn't even know there were purple mushrooms. And if you'd asked me what a mushroom was, I could probably tell you that it wasn't a plant, but that's about all I knew. I was a computer software engineer, and I had never studied botany or zoology or any of the life sciences. But after I photographed this mushroom, I became intrigued with fungi. My view of the forest changed forever. Instead of looking up, I looked down, scouring the ground for hidden treasures. These are just a few of the mushrooms I discovered in those early days. I was very focused on colour back then. This one is Hygrosity graminicola, a stunning green mushroom that grows in the rainforests of Tasmania. It is quite small. It's only 1 to 3 centimetres across the cap. I was very excited when I found this specimen, as they are very hard to see. Moss green mushrooms growing in moss green moss. I knelt down and took the photograph, which I thought was a very special single specimen. And when I stood up, I realised I'd been kneeling in a whole patch of green mushrooms, hiding in green moss. This is a russula, which can often be the perfect looking mushroom. They come in reds, yellows, purples and greens, with bright white gills underneath. They're very appealing to many photographers, and many are edible. Some tasty, some not so tasty, and some not edible at all. This is one of my favourite mushrooms. It's called Mycena interrupta. It appears in April and May on fallen wood in many forests in southern Australia. Stunning, isn't it? All these species are what people generally think of as a mushroom. They have a stem, they have a cap, and they have gills. But then I started to see mushrooms that didn't look anything like this, and I realised I had a very narrow view of what a mushroom really is. Like this orange fungus. It is a species of Ramaria. They come in a dazzling variety of colours. It looks like undersea coral, doesn't it? Which of course attracted me as a photographer. This is one of the many shelf fungus I started to see, Trametes versicolor. These fungi last a long time as they decompose large fallen logs and tree stumps. So they tend to be around even when other mushrooms are not. This one is Cordiarites frondosa. It's an ascomycete, which is commonly called a cup fungus. I never knew that fungus could be such a beautiful black. Incidentally, I've been told that Cordiarites group of mushrooms can be extremely poisonous, so don't be fooled by its beautiful appearance. Many people in China know this fungus. Its name is Hericium coralloides, which we know as the coral tooth fungus. We've seen it in forests in Yunnan, and the locals told us it's an edible mushroom. When I started to discover this huge diversity of fungi, it was like someone opened my eyes. I realised there was much more to mushrooms than just different colours. What started as a photographic journey then became a journey into the science of fungi through the vehicle of photography. My first big discovery was that fungi are not plants or animals, that they are a whole separate kingdom of life. Scientists think there may be 4 to 5 million species of fungus on the planet, but so far we've only documented around 200,000 of them, so there's a lot left to learn. The photographs I take are generally mushrooms, the fruiting body of macrofungi, just like apples on a tree. Some of these fungi are saprobes. These are the recyclers of the forest, where the body of the fungus, the mycelium, a mass of branching thread-like hyphae, grows inside dead wood or leaves or in the soil, breaking down the dead vegetation. Without these saprobes, all the fallen wood in the forest would just pile up, like it did back in the Carboniferous era, over 300 million years ago, when the dead trees were gradually compressed into coal seams. The Carboniferous era ended when fungi evolved the ability to break down lignin in wood and decompose it, recycling all those nutrients back into the soil to feed the trees. Another type of mushroom that I photograph in the forest are called ectomycorrhizal fungi. These live in a mutualistic relationship with trees. They provide the trees with water, minerals, and nutrients from the soil. The trees provide carbohydrates to the fungi. You might have heard of the wood wide web. That's where the mycelium connects all the trees in a forest through a vast network. To a certain extent, the trees can communicate with each other through the web, perhaps warning each other of insect pests or sharing nutrients across the forest. Scientists are just starting to find out how to use this network. But what we do know is that trees connected in the forest by a fungal network survive much better than individual trees do on their own outside the forest. Another very cool type of fungus I've been documenting in forests are the parasitic fungi. Some of these are parasitic on trees and will eventually kill the trees. Some are parasitic on insects. These are commonly called cordyceps. We've found them, mainly in Asia, but particularly in Yunnan in China. We always get a buzz out of cordyceps because they're so difficult to find. You just see the fruiting body coming out of the ground, maybe a little orange stem, and you go, aha, there's a cordyceps. This is one of the fun side journeys I've been on as I've explored the kingdom of fungi. It's a slime mold. Now, slime mold is fascinating stuff, more closely related to malaria and amoebic dysentery than it is to other life forms. It has colourful names like dog's vomit and wolf's milk, but it looks like fungi, so it tends to get lumped in with fungi. And then there are lichen. It's estimated that 6% of the Earth's land surface is covered with lichen. Isn't that amazing? Many people think that lichens are plants, but in fact they're a mutualistic symbiosis between an algae or cyanobacteria together with 2, 3, or even 4 species of fungus. The algae provides carbohydrates to the fungus through photosynthesis, and the fungi's role is to provide the structure to the lichen. Each lichen needs its algae and all of its species of fungi to survive. Everything about lichen is complex, so I'll just stick to photographing their beauty and leave the science to those who are better qualified. The kingdom of fungi is full of cool science like this. Check out this bird's nest fungus. I often find them growing in mulch in my garden, so they're saprobes and they grow on wood. The interesting thing about these mushrooms is the way they spread their spores. You can see that there are little eggs inside the nest. Each of those is actually a spore packet, and the nest is what's called a splash bucket. So when it rains, a raindrop will hit the splash bucket and splash the spore packet out of the nest, sometimes shooting it by as much as 3 meters away. Often there is a fine thread attached to the spore packet. When this hits some low vegetation, that thread wraps around a twig or a leaf, holding the spore packet above the ground. When it opens up and releases the spores, they have much more chance of spreading widely on the wind. It is a very successful means of reproduction. I'd heard that there were mushrooms that glow in the dark, particularly in the tropics. So one rainy, moonless night in summer, I decided to see if I could find them in my backyard. I find a lot of fungi in my backyard. I turned my house lights off, walked in amongst some trees growing near the creek, and turned my torch off. I was prepared to wait for several minutes to allow my eyes to adapt to the dark, because the only luminous fungi I'd seen before were very faint. But as soon as I turned off my torch, there they were, little points of light amongst the undergrowth. I went closer and I found these beautiful little mushrooms glowing a soft, luminous green. It was quite magical, and I spent many happy hours wandering around at night, photographing these beautiful Mycena chlorophyllus. Sometimes they were so bright I could find my way in the dark forest trails with a stick covered with the mushrooms. Finding these has opened up some incredible doors to sharing the story of fungi. They were the first mushroom that I time-lapsed. To my surprise, my wife supported the idea of filling our spare shower with rotting logs, blacking out the windows, and turning it into a time-lapse studio. But then Catherine is a filmmaker. The results surpassed our expectations. My time-lapse became more ambitious, and I set up 2 studios, one in a shed and one in a shipping container. When the BBC saw the movies I created, they fell in love with them. They were showcased on Planet Earth 2 with David Attenborough, and this sequence was listed by the BBC in their top 10 clips from that series. Since then, time lapses of Mycena chlorophyllus and many other species of forest fungi have been included in 10 other international natural history documentaries. Tom May, a mycologist or fungi scientist at the Royal Botanic Gardens in Melbourne, has been very supportive of our work. He says that the time lapses bring fungi alive, like a mammal. For the first time, ordinary people and scientists can see things they've never seen before. In Australia, there are only a handful of mycologists, and it's challenging for them to be in the forest when the fungi fruits. So, mycologists became very interested in what my photography and the time lapses could show them about the fungi. And my role as a mere fungi photographer shifted to a role as a naturalist and a collaborator with the scientists. Just like the naturalists of the 1800s, I had the equipment, the passion for the natural world, and the time and opportunity to explore. I became interested in not just capturing the beauty of mushrooms, but also capturing the form and the structure with a scientific accuracy that complemented the research of the mycologists. Some of the mushrooms we find are just so different that there is little doubt that we've discovered something new. The blue was like that. I first found it about 10 years ago, quite close to my home, in this forest in fact. At first I wasn't sure it was a mushroom at all. I thought it was maybe a blue sweet wrapper on the ground. It was just so blue. I sent samples to Dr. Tom May, and he identified it as a new species, similar to specimens first documented in New Caledonia. Now, a decade later, the original single species from New Caledonia has been split into 3 species, which look similar but not even closely related to each other. And this blue mushroom still hasn't been given a name. I think it's a fascinating fungus, and Dr. Tom May called it the most beautiful mushroom in Australia. I always try and get a time lapse of it, and sometimes I succeed. Scientific collaborations like this one was the next significant stage of how fungi changed my view of the world. In 2014, I received an email from Dr. Peter Mortimer, who is a professor at the Kunming Institute of Botany, which is part of the Chinese Academy of Sciences. Peter and the head of department, Professor Xu Jianchu, led a team of mycologists who are doing some extremely exciting fungi research. Peter's dad in South Africa had seen my photographs on the internet and suggested that he contact me. The outcome of that first email has been 4 trips to Yunnan to photograph and document the fungi of the remote forests. It was on these field trips that I realised just how edible fungi can really be. In Australia, we're what's called a fungi-phobic society. In our shops, you can find only about 5 species of mushroom for sale, and they're pretty boring. People are generally scared to eat forest fungus. We just don't know which ones are poisonous and which ones are edible. But in Yunnan, we discovered that people eat around 900 species of fungus. Definitely a fungi-philic society. I have to confess that at home, I don't really like to eat mushrooms. But in Yunnan, we were introduced to many delicious mushrooms, and local villagers shared their knowledge about what was edible and how to cook them. So this is a Belita's, this one is edible? You eat this one? This is very good.
It's a kind of typical delicious eatable mushrooms. Volunteers do this.
This is their favourite one to find. We also discovered that mushrooms in China are a huge business. It was in the forests of China that we started to understand just how interdependent the trees and the animals and the fungi really are. My view of the world was changing, yet again. The forest was starting to make sense. It wasn't just a bunch of trees. Even in a relatively simple forest, there are probably hundreds of thousands of life forms, all dependent on each other and all contributing to make up what we call a forest. From viruses and bacteria through to fungi, plants and animals. They are all part of the huge intricate web of life. And the fungi are a really important part of that ecosystem, and yet we know so little about them. On the China field trips, 1 in 7 species that we document is new to science. But even the ones that are known are truly fascinating. Here are a couple that I found really exciting. These are called Tometomyces, and I first photographed them in Yunnan. They have a hard, pointed cap which allows them to push through the ground from up to a metre below the surface. They live in a mutualistic symbiosis with termites, which means that neither species can survive without the other. The termites collect grass and wood and take it underground to their nest, where they feed it to the fungus. They then eat part of the fungus. The fungus eventually fruits, pushing the mushrooms up above the ground. Tometomyces came as a complete surprise to me. In Australia we have plenty of termites, but no Tometomyces. Yet many species of this fungus are found throughout tropical Asia and Africa. There is one species of this fungus that produces mushrooms up to a metre across. Many Tometomyces are valued as a delicious edible, and we often see them in markets across this region. We certainly like them. This little purple mushroom is called Lacaria amethystina, and it grows mutualistically with pine trees. Villagers in Yunnan prize the mushroom as a pretty edible that looks good in any dish. I just love that it is such a beautiful colour. The most poisonous mushroom in the world is an amanita, Amanita phalloides, more commonly known as the death cap. They grow under oak trees all over the world, and every year people die from eating this mushroom. For instance, this amanita we documented in Myanmar we think is part of the Amanita phalloides group. It may be the source of a number of recent mushroom deaths. The mycologists we were working with and village leaders were keen to document it, to warn people against eating it. But this is a very similar Amanita mushroom that people in Nepal say is edible. There isn't a lot of difference between them, is there? Just to fill you in on what happens if you eat an Amanita phalloides. It is actually reported to taste quite good, but I really can't verify that. The first symptom is that you feel a bit sick the next day. After 3 or 4 days, you are so sick that you know you need to see a doctor. But by then, it is often too late. Amanita phalloides contains a poison that destroys our liver, so slowly over several weeks, we die. This is a mushroom that's very well worth avoiding. So there's no doubt that photography can play an important role in educating people about these mushrooms, and other poisonous mushrooms too, as is its role in educating people about the fungi they can eat. The China collaboration has led to similar collaborations in Nepal, Myanmar and India, working with organisations focused on conservation and helping local people manage their forests in a sustainable manner. Often as roads are put in, forests are cut down, much to the detriment of the local people. edible forest fungi that could be used for food or trade often vanishes with the forests. So the photographs have been used to produce a number of field guides for villages in these countries. But for me, the most exciting part has been to document all of the fungi in these remote areas and learning a tiny bit of how each one interacts with the forest. And the fungi in the eastern Himalayas has been absolutely superb. In the last year, our role as fungi educators has taken on an even wider vision. On these international fungi adventures, we are often introducing our collaborators to fungi for the very first time. We watch with joy as they become excited about this world that they had never noticed before. Every time, we see them become almost as passionate and obsessed as we are. And we want to see if we can make this happen on an international scale. So in India in 2018, we filmed our whole fungi safari. Here's a little taste of what we did. This is a scene where we document, for the very first time, a luminous fungi in Meghalaya. A fungus species I time-lapse that never ceases to wow is Mycena chlorophyll. It's a very bright, luminous fungus that I find in my local forests. Wherever we go, we always ask if there is a local variety. Usually the answer is no. So do you have any mushrooms here that glow in the dark?
Yes.
You do?
Yeah.
So do you find many of them?
Yes, of course.
What do you call these mushrooms?
This is called bright mushroom because we give out the light in the night time.
We go down and find some?
Sure, I'll contact you.
That's brilliant. Wow, look at these. They're nothing like the fungus we get at home. The stems glow, but the caps don't glow, whereas at home, the caps glow and the stems only glow a little bit.
Ah, so it's same but different species.
Same but different, yes. You really can't see these with the light on them, but we'll get them up in the dark and I can photograph them with long exposure and then you'll really see them in their full glory. There are currently around 80 species of luminous fungus recorded on the planet, but only a handful of them glow as brightly as this one. This is the first time Malinong's luminous fungus has ever been documented, and when its DNA was analysed, we discovered it's a new species. Now I'm going to take you back to my home in Australia. In 2019, we experienced one of the worst bushfire seasons we had ever seen. 17 million hectares of land was burnt, over 5 million in my state of New South Wales alone. It was very frightening and destructive, and particularly scary when it started burning the rainforest near where we live. Days after the fire had been through, we started hearing reports from firefighters that mushrooms were appearing in the ash. So we drove up to the blackened landscape to see what we could find. We were amazed. There were lots of fungi, and they were species that we had never seen before. This one is what's called a stonemaker fungus. It started to appear a day or two after the fire, while the ground was still hot and smoking. It lives in the earth for many years and forms a hard mycelium lump underground, which resembles a stone, hence the name. It only fruits when there's fire, which in this particular forest may happen only once in 100 years. Then we saw masses of tiny cup fungi covering the ground. We managed to identify them as Anthracobia muelleri. We started to wonder about its role in protecting the forest soil, as fire tends to make the soil very fragile and easily washed or blown away. But this fungus appeared to be binding the surface soil together and locking in any moisture. For me, these fungi raised so many questions. Where does this fungi live when there's no fire? It has no deep mycelial reserve that I could see. I'd read that there was a similar fungus in North America which lived as a microscopic fungus in the cellular structure of mosses. Perhaps this did something similar and only became a macrofungus when the fire had sterilized the soil of all competing organisms. We even found mushrooms in gaps underneath charcoal, where there had once been fallen logs on the forest floor. This would have been where the fire was its very hottest. How could this happen? How could spores make their way into the soil that had been completely sterilized by the fire? The variety of fire-loving fungi was astounding. And then when we went back a month later, there were different fungi coming up. We published a few videos and the response surprised us. Many of the locals told us they were uplifted by this positive information in amongst all the news of destruction. They were heartened to hear just how resilient the forest could be, thanks in this case to its fungi. Life is such a wonderful thing and we know so little about almost all of it. As I told you at the start, I used to work as a computer software engineer on very large computer systems, and systems that we think of as being very complex. But a computer system is designed by humans, and get enough of us together and we can explain everything about them. But even the smallest system in the natural world is more complex than the largest computer system. Then when you consider that there are trillions or even quadrillions of organisms on the planet, we start to get a sense of just how complex life on Earth really is. I started on this journey knowing a little bit about photography, but very little about the natural world. Learning about the complexity of fungi and through it the complexity of life, I realize now that life on this planet is more interconnected than I ever could have imagined. We are just one organism in that story, and yet we have the means to destroy it all, including ourselves, because we cannot survive on our own. With all the extraordinary tools we have created, humans have a wonderful opportunity to learn and conserve. One of the scientists we collaborated with in India said, "You can't conserve a forest ecosystem if you don't know what's there." And I feel we have been handed this wonderful opportunity to help document a very small part of this complexity and share it with you. From one photograph of a very small purple mushroom on the ground, I've been taken on a journey that reveals the very big picture of life on this planet. Thank you.
Okay, class, hopefully you guys have enjoyed this video, and then we shall see you in the next class.